Dear Chairman, ladies and gentlemen, in the next short seconds, or minutes, I would like to introduce our research group in the Institute of Surgical Research, and I have to take it instead of Professor Boros, who has participated in the other conference. So at the moment, I am standing of Professor Boros. So in the medical practice, day by day, have to a real battle with the disorders, mainly the circulatory disorders. So in this battlefield, there is increasing significant role of the on the animal experiment, animal models, which are used to study to the basic mechanism of the medical problems and developing new tools for diagnosis and therapy. With this object, our institute deals with these animal models, and we are used the wide color spectrum of the pathological event. And we are focused from the reactions to the different type of circulatory shocks. So we measured several physiological functions, and we are monitoring the cardiac and pulmonary function. We are focused to the gastrointestinal function because the gastrointestinal tract is an absolute sensitive area of the certain disorders, and mainly we have a special device and image techniques to monitoring the microcirculation and examine the mitochondrial function. So the next slide summarizes those therapies which are used regarding the studies. Nowadays, we are focused specifically to the biologically effective gases. As a well-known gas is a nitric oxide, but it's absolutely new an idea to study the methane. What is the methane? So relatively it is an inactive gas, but it results from the composition of organic matters in anoxic environments. I would underline. There are several principal sources, natural sources as marshes, but it can be realized, it can be detected in the digestion system in humans, animals, both. And in vivo formation, it's actually attributed to the metagenic archaeobacteria. So we have three different ways to study the methane. The first is the endogenous production of the methane. Secondly, we focus on the exogenous therapy with methane. And nowadays, the newest way to examine the diagnostic possibilities with methane. What does it mean shortly? Endogenous methane production can be occurred under reductive stress. That is agents increasing and can be influenced to these membrane components which have a methyl groups. It's rich in methyl groups, mainly the phosphatidylcholine. This choline component have a relatively rich the methyl groups, and the methane can be produced from these methyl groups and increase as a free gas and can influence several procedures. So as exogenous methane, in opposite to the previous line, we can use the therapy with a 2.2% methane, and surprisingly, we can have a very significant results to influence the inflammatory reactions and ameliorate the mitochondrial dysfunction. Several words, the new diagnostic way. So it's a methane can be produced by the bacteria flora in our intestinal system, or we can administer with methane this intestine, and through this microcirculatory system, spongic circulation, pulmonary system, through the gas exchange through alveoli, the methane it's exhaled and measured, so we can calculate or estimate to the rate of this microcirculation based on the measured exhaled methane. So I would like to introduce and our students, our talent students, our senior students, Naszai and Liliána will demonstrate in their first steps in this new area, their first results regarding this methane effective therapy. So thank you for your attention, and I would like to give the say to the first speaker in our institute. Thank you very much.
Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank you very much. Thank So, dear audience, dear guests, our research work is focused on the smallest organic molecule, the methane. It's non-toxic and biologically inactive, but in different conditions, it can be released in bacterial and non-bacterial organisms. Some data suggest it could be a gas transmitter in biological systems and do hint at an association. With a small vowel motility regulation. Methane also has a tissue protective effect. In these canine intestinal mucosa sections, you can see the positive effect. After 1 hour of ischemia with 3 hours of preperfusion, significant denudation of villi is observed. With methane treatment, the mucosa is almost as intact as sham-operated animals. But what is the reason of this incredible tissue damage? During oxidative and nitrosative stress, reactive species such as hydrogen peroxide, superoxide, and nitric oxide releases. What's more, superoxide and nitric oxide forms peroxynitrate, which is an even more reactive agent. Our hypothesis is that methane has a protein conformation modifying effect through which it influences enzyme activity, and methane possibly reacts with nitrous species such as peroxynitrate. Our aims are to find biochemical targets where methane can influence the mechanism of oxidative and nitrosative stress, and to find components which can be involved in anti-inflammatory action of methane. And our possible biochemical target is xanthine oxidoreductase. In normal oxygen tension, xanthine oxidase oxidizes xanthine into uric acid using its +6 molybdenum cofactor, and it releases hydrogen peroxide and superoxide. In the case of lack of oxygen, for instance during hypoxia, and increasing reductive agents such as NADH, xanthine oxidase mainly uses its +4 molybdenum cofactor to reduce nitrate ion to nitric oxide. As you can see, this enzyme can produce the elements of oxidative and nitrosative stress. What's more, superoxide and nitric oxide forms peroxynitrate, an even more reactive agent. Our methods were the following. We mixed xanthine oxidase enzyme, nitrate ion, and NADH into the reaction mixture, which we incubated at 37 Celsius degrees. The bubbling lasts for 10 minutes, and we treated this mixture with normoxic air with or without methane. The methane concentration was 2.5%, and it cannot be higher regarding to the methane's explosive nature. After the reaction, xanthine oxidase activity was measured by fluorometry, reactive oxygen species by chemilumiноmetry, and nitrogen concentration by photometry using Rees reaction. The next slides demonstrate the results, and the figures represent the mean and the standard deviations of data. Our first measurements our measurements for the first hypothesis were about xanthine oxidase activity. Methane treatment diminished the xanthine oxidase activity. We could not prove the pH dependency of xanthine oxidase, but in 3 cases, the difference proved to be significant. The chemilumiноmetric measurements shows decreased reactive oxygen species production. The luminal enhanced chemiluminescence proved to be significant. The next slide shows the nitrate concentration changes after methane treatment. Methane diminished the nitrate concentration. We think that this decrease in nitrate concentration means a possible increase in nitric oxide formation. Sadly, up till now, we could not measure this other side of this reaction, and we are trapping a nitric oxide. We are planning a nitric oxide trapping experiment. We tried to prove our second hypothesis, the reaction of tyrosine with peroxide nitrate. We treated these mixtures with normoxicare with or without methane. Nitrotyrosine concentration was measured because this is the stable end product of this reaction. Our preliminary results shows that methane decreased the formation of nitrotyrosine, but it's just preliminary, so we have to do other examinations. So our conclusions are the following. Exogenous methane decreased xanthine oxidase activity, reactive oxygen species, and nitrate concentration, which can mean an increased nitric oxide production. And more studies are needed to specify peroxide nitrate and methane reaction and physicochemical changes of proteins. And we want to see whether methane has an effect on other enzymes, for example, nitric oxide synthase enzyme. And I want to thank you for my mentor and for your attention as well.
Dear Chairman, dear audience, in my study I would like to focus on in vivo effects of methane inhalation. Firstly, the tissue ischemia is a frequent consequence of circulatory disturbances such as occlusive ischemia, non-occlusive ischemia caused by hypovolemia of cardiogenic shock, or transient ischemia. The different tissues have a different tolerance of hypoxia. The most hypoxia-sensitive tissues are the following: intestines, lung, and tissues of the central nervous system. The reoxygenation is necessary for surviving tissues, but it's a double-edged sword because it initiates complex biochemical cascade reactions, and it leads to oxidative and nitrosative stress and tissue damages. But what is the background of this process? Under hypoxic circumstances, have decreased amount of oxygen, which leads to mitochondrial dysfunction, ATP degradation, and hypoxanthine accumulation. The xanthine dehydrogenase conversion to xanthine oxidase is a consequence of hypoxanthine accumulation caused calcium accumulation, and the xanthine oxidase degrades hypoxanthine to xanthine, and after xanthine to uric acid, and forms reactive oxygen species, like superoxide radical. The reactive oxygen species cause peroxidation of membrane lipids with oxidative stress. The other way, the product of nitric oxide synthase enzyme reacts effectively quickly with superoxide radical to form peroxynitrate. All in all, the oxidative and nitrosative stress cause structural and functional damages, and this leads to tissue damages. But how can we moderate these stress reactions? The answer could be the methane. The previous experiments at the Institute have shown that the ischemia reperfusion caused tissue damages can be decreased with methane inhalation. Here in this picture we can see the ischemia reperfusion caused tissue damages without methane treatment, and here in this picture we can see the small intestinal mucosa with methane treatment. And here is the control small intestinal mucosa. Our hypothesis to explain that the methane reduces oxidative and nitrosative stress response in vivo, and with this subject we examine the in vivo effects of methane on xanthine oxidoreductase activity and nitric oxide production in the intestinal and lung tissues under hypoxic conditions. The experiments were performed with SPRD mild anesthetized rats, and the tracheostomy was performed to facilitate spontaneous breathing and respiration. The right jugular vein was cannulated for the infusion, and the right common carotid artery was cannulated for the mean artery pressure and heart rate measurements and the 2 blood samples. Here I would like to say a thank for the assistance of the Institute of Surgical Research for the surgical preparation. And after the preparation, mechanical ventilation was started with a rodent respirator with normal L for 30 minutes. And then the respiration was continued with different gas mixtures. The animals were randomly assigned to 2 groups, and both groups were ventilated with oxygen-depleted air with or without methane. And at the end of the inhalation procedure, we took tissue samples and blood gas samples. And the measurements, the arterial blood samples were analyzed for partial pressure of oxygen, carbon dioxide, and pH and base access immediately post-sampling with an automatic blood gas analyzer. And the xanthine oxidoreductase activity was measured by fluorometry. And we measured the all nitrate content changes because these are stable end product of nitric oxide. We measured it with photometry using grease reaction. And the free nitrotyrosine level as a marker of nitrosative stress and peroxynitrite production was measured by enzyme-linked immunosorbent assay. This table shows that the methane inhalation did not exert significant effect on the blood gas parameters. The effect of methane inhalation on xanthine oxidase activity, we measured it in 3 parts of the intestine and in the lung because these are the hypoxia-sensitive tissues. And the methane inhalation significantly decreased the xanthine oxidase activity in the ileum, in duodenum, and in the lung. The nitric oxide production was measured by detect all nitrate content changes. And we can see that the methane significantly increased the nitric oxide production in the lung. And finally, we measured the nitrotyrosine production because it's a marker of tissue damage of a peroxynitrite production. And we can see that the methane significantly decreased the nitrotyrosine production in the lung. And based on the measurement results, our conclusions are the following. The methane is able to decrease xanthine oxidase activity significantly under hypoxic circumstances. And the methane increased nitric oxide production in the lung and colon, but not in other intestinal tissues. The methane decreased the nitrosative stress response in the lungs significantly. All in all, the methane decreased tissue damage through influence decreases xanthine oxidase activity and increases and influence nitric oxide production. And finally, I would like to say a thank for my mentor and thank you for your attention.